This video is going to look at the 3 stages of respiration, and it's also going to look at fermentation in animals, plants and yeast. This is intended to be a really quick overview of quite a wordy process, so you might need to watch it a couple of times, particularly to learn certain definitions and remember the key words. Okay, so looking at stage 1 of aerobic respiration, we're looking at glycolysis, and this is going to occur in the cytoplasm. This stage doesn't require any oxygen. We're breaking down a 6-carbon molecule of glucose into 2 3-carbon molecules of pyruvate, and in order to do that we need to invest some ATP. So the first stage that we're going to look at is called the energy investment phase. What we are doing is we are going to phosphorylate glucose and intermediates to drive forward this reaction. And to do that, we need to invest 2 ATP. And if we look at a molecule of ATP, we can see that it is made up of 3 phosphates. All we're going to do is we're going to break the bond in between that phosphate there, and we're going to give the phosphate to glucose and intermediates. We're going to phosphorylate it. And when we do that, we produce 2 ADP, and you can see that just looks the same as our ATP molecule, but it's missing that phosphate. So how do we recognise the energy investment stage? ATP is used. And that investment is going to pay off in the energy payoff phase where we are making ATP. We're going to get this net gain of ATP. So here we have 4 ADPs. So we can see it looks the same as our ATP molecule, but it's missing that phosphate. And what ADP is going to do is it's going to steal back those phosphates. We're going to phosphorylate ADP to make 4 ATP molecules. We can see that we've got that last phosphate that we've attached to our ADP. So ATP is made. We get that net gain. The other thing that is happening in glycolysis is we have these enzymes called dehydrogenase enzymes. And what their function is they remove hydrogen and electrons from these intermediates that we have in between glucose and pyruvate, and they give them to this coenzyme called NAD to form NADH. And what NADH is going to do is it's going to pass those hydrogens and electrons to stage 3, the electron transport chain, which we'll talk about a little bit later. Okay, so the next stage is called the citric acid cycle, and this is located in the matrix of the mitochondria. Now, this stage is only going to occur if we have oxygen available. We'll talk a bit more about what happens when oxygen isn't available later on. So this pyruvate that we made in glycolysis, this is going to be broken down into something smaller. We break this down into a 2-carbon acetyl group. And you can see here we've gone from a 3-carbon molecule to a 2-carbon molecule, so we're going to release carbon dioxide here. We also still have our dehydrogenase enzymes that are removing hydrogens and electrons from intermediates, and they're giving them to coenzyme NAD to form NADH, and that can just be taken to the electron transport chain. Our 2-carbon acetyl group is going to join with this other coenzyme here. It's going to join with coenzyme A, and when it does that, it forms a 2-carbon acetyl-CoA, or a 2-carbon acetyl coenzyme A. And then we can start this citric acid cycle. So the 2-carbon acetyl coenzyme A is going to join with 4-carbon oxaloacetate. We've got a lot of long words here that we need to remember. And when that 2-carbon molecule joins with the 4-carbon molecule, we form a 6-carbon molecule, and that molecule is called citrate. And through this series of enzyme-controlled steps, we convert that back into oxaloacetate so we can continue to do the citric acid cycle. And we need to be able to say the 3 things that are produced in the citric acid cycle. We're going from a 6-carbon molecule to a 4-carbon molecule, so again we're releasing carbon dioxide. We're releasing a little bit of ATP, and we also have our dehydrogenase enzymes playing a key role here, so we're releasing NADH again. Okay, so the last stage that we need to look at is the electron transport chain, and this is located in the inner mitochondrial membrane. I've drawn a little diagram here of the mitochondria. We can see that the citric acid cycle is in the matrix of the mitochondria, and the electron transport chain is along this inner mitochondrial membrane. And we can see here, if we look at the electron transport chain, we can see that it is made up of these things called carrier proteins, which we can see here. And we also have this molecule here called ATP synthase, which is our enzyme that is going to produce lots of ATP. And we've been talking a lot about NADH from these previous stages, and they're going to pass those hydrogens and electrons to the electron transport chain. And those electrons are going to pass between these carrier proteins. And when they do that, they are going to release energy. And this energy is going to allow us to pump this hydrogen across the inner mitochondrial membrane. And when they reach this molecule called ATP synthase, the hydrogens are going to flow down through the ATP synthase. The ATP synthase is going to spin and it is going to produce this burst of ATP. So I think of it a bit like a turbine in a dam generating electricity. And finally, these hydrogens and electrons are going to combine with oxygen to form water. And oxygen is essential for this process here. It is the final hydrogen and electron acceptor in this chain. Okay, so starting to look at what happens when oxygen is absent, we have this process called fermentation. And fermentation allows us to continue to make a little bit of ATP when oxygen is absent. This is going to occur in the cytoplasm. Because remember, we can't do any citric acid cycle, we can't do electron transport chain because those require oxygen. Remember, oxygen was our final hydrogen and electron acceptor in the electron transport chain. So how is this happening? Remember, when we have glucose being broken down into pyruvate, we have our dehydrogenase enzymes that are removing those hydrogens and electrons from intermediates, and they're giving them to coenzyme NAD to form NADH. And usually that NADH would be taking the hydrogens and electrons to electron transport chain, but that pathway can no longer occur. So what NADH does instead is it gives those hydrogens to pyruvate to form something called lactate. So this might be happening when we're doing really intense exercise or something like that. We're not getting enough oxygen into our cells. But what this allows us to do is it allows us to regenerate NAD. We can keep accepting more hydrogens and electrons. We can keep breaking down glucose into pyruvate to release this little bit of ATP. And when oxygen becomes available, we can convert that lactate back into pyruvate and continue with aerobic respiration. So how are we forming that lactate? NADH is giving hydrogen to pyruvate. And why are we forming that lactate? It is so that we can regenerate NAD, glycolysis can continue, and we can keep making more ATP. So just looking at fermentation in animals as a simplified diagram, we can see that pyruvate is being broken down into lactate in animal cells. And once we have oxygen available to us again, we can reverse that process. So lactate can be converted back into pyruvate. We continue with aerobic respiration. So the products produced in fermentation in plants and yeast are different to animal cells. This time pyruvate is converted into ethanol and carbon dioxide. And when we've made these products, they can't be converted back into pyruvate like in animal cells. We say that this process is irreversible. And I always remember the products produced by thinking about beer being fermented using yeast. It's fizzy because of the carbon dioxide being produced, and it contains alcohol because of the ethanol being produced. So overall, fermentation allows the cell to make a little bit of ATP when there's no oxygen, but much less than aerobic respiration.